So the first hour was spent with a very superficial survey of what I consider some very basic properties of thalamus and cortex. And I'm going to use the next hour to sort of get into more complicated stuff that I find more interesting. It's going to be kind of random. There's not much organization to this. And I don't know how far I'll get. And some of the questions that were raised in the first hour, I might be able to provide some answers to if I get there. So first topic has to do with interneurons and thalamus. They're quite an interesting cell type, and I think hope to demonstrate that to you. And they are organized in a particular kind of synaptic arrangement that are called triads, and that's what I want to explain. So if we look at the cell types typically that are found in thalamus, this is your relay cell, big, healthy-looking cell with a fairly large cell body. And somebody asked the question afterwards, and I forgot to mention it. The relay cells throughout thalamus outnumber the interneurons, that's what this guy is, by about 3 or 4 to 1 throughout thalamus with some variability. So these are about 75 to 80%, and these are the rest. Okay, so this is the thalamic reticular cell, GABAergic. This is the interneuron. And the relationship between these two becomes quite interesting. Now the interneuron...
Can you remind again, please, what a relay cell do?
I'm sorry?
What does relay cell do?
Relay cells are the cells that get input. If this were a geniculate relay cell, get an input from the retina and project up to cortex. Okay. The interneurons do not. They only innervate these guys. Okay, and the innervation of relay cells comes from a traditional axon that provides a terminal output that's known as F1. The terminology, there's a reason for calling them F1s, but let's not go there, it's not important. Just that they have a particular kind of output from the axon that's fairly traditional. This is a typical kind of axonal output you find in GABAergic cells in cortex as well as thalamus. What's interesting is their dendrites, here's a blow-up, also have these bulges that look like synaptic terminals. And what we'll see is that most of the outputs of an interneuron, synaptic outputs, come not from the axon, but from the dendrites. Here's a stain of an interneuron, and these dendrites look like axons with terminal arborizations. If we take a blow-up of this area here, this looks like an axon, but it's actually a dendrite with these clusters of appendages that look like presynaptic terminals that you'd find in an axon. And in fact, they are presynaptic terminals. So if we look at the synapses that come from these dendrites, and by the way, I want to make clear how unusual this is, because if we go back to the neuron doctrine and the idea that neurons are polarized, that the inputs come into dendrites, get integrated, and the axonal output is the only output. This defies that. This is a very different kind of cell where most of the outputs are actually coming from dendrites. So this is what one of these dendritic outputs are doing. And they engage in very curious kinds of synaptic arrangements. And there are a couple things about this I want to emphasize. And I'm using as a first example the kind of triad we see in the lateral geniculate nucleus. You get a retinal input. And there are 3 synapses involved here. The retinal terminal makes a synapse onto a relay cell dendrite, but also onto one of these terminals from the interneuron. And the interneuron then makes the 3rd synapse onto the same process that the retinal terminal does. That's why it's called a triad, 3 synapses. So you get direct excitation here and indirect inhibition through this route. Is that clear to everybody? There's an important variable here. You notice that this is glutamatergic, and it's activating only ionotropic receptors, mostly AMPA receptors here. But here it's activating metabotropic glutamate receptors. So even though it synapses on both structures, because of the receptors, we're going to see there's an important functional distinction here. Now this is the case in the LGN. If we picked any thalamic nucleus, we just take the driving axon, whether it could be medial lemniscus if this were somatosensory and so on. You get the same kind of synaptic arrangement. I have this sort of purple cloud here. We're calling that the glomerulus, and I'm going to explain why that's also of interest, although very poorly understood. If we look at typical synapses anywhere on a dendrite, in cortex and thalamus anywhere, each individual synapse is encapsulated by a glial sheath. And the role of the glial sheath is not entirely clear. It was thought, among other things, to prevent spillover of the transmitter that's released from this terminal to affect receptors elsewhere, that it soaks it up pretty quickly. But it probably plays other roles as well in synaptic transmission that are just being investigated. But the point is, typically every single synapse has this local glial sheath. But in glomerulus and thalamus, and we don't see this in cortex, these individual synapses are naked. There's a glial sheath around the whole thing. And what this may mean possibly is that a transmitter that's getting spilled out from these terminals can spread much further and have more effect. Beyond that, this has been a mystery for decades. People have tried to figure out what the functional significance of this difference is, but we really don't know beyond just guesswork like I offered. So something is going on here that we don't understand. And I won't have more to say about that because we don't know anything more about it. Now the interneuron itself is quite interesting then, and we think it's multiplexing. So we have on these terminals that are both pre and postsynaptic, which makes them pretty unique to be a synaptic terminal that both is presynaptic to the output to relay cells, but also postsynaptic to a variety of inputs, not just retinal input or the equivalent, but also local axonal inputs from the brainstem reticular formation and the axon of the interneuron, of other interneurons in these glomeruli. And proximately, there are inputs to the proximal dendrites from retina and from other inputs, mostly from brainstem. And this is the way we now think. This is a very unsophisticated model for how the interneuron is functioning, that the proximal inputs are controlling the axonal output because these produce EPSPs and IPSPs that are going to control the firing of the axon, and that's your traditional organization. But each of these represents a local circuit, and because of the very thin dendrites that they emanate from and how distal they have very little influence on neighboring ones and no influence or very little on the cell body. So we're multiplexing hundreds of local circuits out on the dendrites, and at the same time, the same cell supports an axon with more conventional dealing with inputs. So this is what we think the way the interneuron is functioning. Any questions about that?
Two separate systems?
Yeah, completely separate as far as we can tell. The modeling suggests that. There's some experimental evidence that does show that if you record from the cell body and you can activate these chemically, you're not seeing any evidence for that there. They do seem to be electrically isolated.
What about the spike? Can the spike propagate out to
Yeah, that's a good question. And when I say those experiments are done in slices, the possibility that there's some active conductances in these dendrites is there. They haven't been demonstrated. But it may be because these active conductances can be turned on and off by modulatory inputs, that you don't see them in slices and only see them in real animals. So the possibility is that this could have some effect here under certain conditions where modulation is allowing them to happen, but that's pure speculation. Yeah.
I understand cerebellum also has this kind of glomeruli of three. Are they analogous?
So they're glomeruli.
What is, no.
I don't know about the cerebellum, but the olfactory bulb has glomeruli as well. They're completely different. It's unfortunate that they have a similar name. I think glomeruli is a term that's used for complex synaptic organization, and that is very confusing. But these kind of glomeruli, as far as I know, don't exist anywhere else. Yeah.
So what if they were all being modulated by different gamma frequencies, and then the cell in the middle could listen to the one it wanted to?
Whoops.
By tuning in.
These are somehow oscillating.
I think gamma's on all the somas of all the cells in the thalamus.
We're not seeing much evidence of that in our recordings. And I know that these oscillations have been reported and do exist, but under the conditions we're looking at in our behaving mice, we're not seeing it. So as a result of that, because we don't see it, I can't tell you much about it. Doesn't mean it's not there and doesn't mean it's not important, but we don't have a handle on it. Okay, so here, I'm trying to figure out how to explain this, what we think the triad is doing. and this may not make much sense, but it's based on the fact that when the retinal axon is firing, it's activating ionotropic and metabotropic receptors. And remember, activating the ionotropic receptor is done at even low firing rates, but you're not going to get this one activated until the firing rate gets elevated. So we can imagine when the triad is not active and there's a contrast response function, that the contrast of a visual stimulus is monotonically related to the firing rate of this axon. That when there's not much firing here, we get a contrast response function that looks like this, because the relay cell is getting, at very low levels, just input that's from the relay cell. And because the metabotropic receptor here isn't activated, this circuit is shut down at low levels of firing here. So you get a very sensitive contrast function. But as we increase the firing of the retinal axon, we can start to bring in more and more of this circuit. And when you do that, you're going to, as you increase the firing here, you're going to increase inhibition here, and you're going to make this whole process less sensitive, and you have basically adaptation of contrast here. So higher contrast, leading to higher firing of the retinal axon, will bring in more and more of the inhibitory circuit here because of the activation of metabotropic receptors requiring higher activation levels. And then if you were to shut this off, that contrast, before the contrast sensitivity reverted back to its pre-level, would take about 3 or 4 seconds. That's because once you activate the metabotropic receptor here, it's been shown in this system that the activation lasts for about 4 seconds. Remember, these EPSPs, in this case it is an EPSP, can last for several seconds to several hundred milliseconds to several seconds. And here it seems to be 2 to 3 seconds. So this entirely fits what we know about contrast adaptation. And evidence for contrast adaptation to allow you to be sensitive to the contrast range in your environment with this kind of time course has been demonstrated for retina and cortex and the thalamus, and we're suggesting this is one way it could happen in thalamus. Now this is complicated, I'm not sure I did a very good job of explaining it. But any questions about this? This is just trying to put some functional significance to this curious triadic arrangement that's found throughout thalamus, including human thalamus. Yeah.
Contrast adaptation?
Sorry?
Contrast adaptation,
Yeah.
exactly.
So when you're in a low contrast environment, your sensitivity contrast is pretty high. If you went from this room to a desert at noon, the contrast is much higher. You want to be less sensitive. And this tells you how it could happen, because your retinal input would be stronger, it would activate the inhibition here more, and it would reduce the gain of retinogenicular transmission.
Just one question about that. I mean, if you just wanted to reduce the contrast response, it seems like you could just turn down the gain of the neuron. You could just sort of act on the whole relay, the relay neuron itself. Whereas what's interesting about this triadic arrangement, I think from a computational point of view, is that you can, you're modulating the information synapse by synapse, right? It gives you a very fine-grained control that maybe you could do something else.
If you try to reduce the gain here by inhibiting the neuron globally, you're affecting all the synapses. The nice thing about this is it's affecting only the retinal input, and that's what you want for contrast adaptation, I think. And it leaves the other synapses unaffected. Their gain is unaffected. So if you want to modulate or do other things, you're not interfering with that.
The other synapses from like brainstem or some other place.
Or the cortical feedback. Okay. That's what I have to say about triads and interneurons. I'm now going to talk a little bit about parallel processing, which is an important feature of at least sensory processing. It applies to all our sensory systems. And this was hinted at already in previous talks, particularly Jeff talked about this. If we look at ganglion cell types in the cat, we have different kinds of ganglion cells that have very different morphology and very different physiology. The main ones, the first ones described were called X and Y. This is why the label here has been lost for X, but they have different receptive field properties. The Y cells have larger receptive fields, are more interested in motion, in lower spatial frequencies. These seem to be more dedicated to fine details and less interested in motion. And what this says is that at very early levels in retinal processing at least, we divide up information in the visual seeing into different channels. And this makes more efficient processing of the information. We also have a third cell type called W. Again, the X is missing here. And this has a garbage bag of cell types. It probably, the W cell was, when it was first defined, was thought to be a unique cell type. But now we think there's many as 10 or 20 different cell types subsumed under this classification. And that's still being worked out. These are pretty homogeneous each. But these include cells that have the classic center-surround organization, as you see in X and Y cells. Other cells have diffuse on and off zones that aren't organized in a center-surround. Some of them show direction selectivity, and some of them show color sensitivity. So there's a wide variety of different cell types that we're lumping together as W. But these represent separate channels that come out of the retina. And we see the same thing in the monkey. The M cell, called M cell because it innervates the magnocellular layers. The P cell innervates the parvocellular layers of the LGN and the coniocellular cell. And these are equivalent, homologous specifically, to Y for M, X for P, and conio is equivalent to W, and also has a number of different cell types. So we get these separate channels coming out of the retina, and in the cat, they're organized in the following way. The X and Y cells innervate the A layers. It's a laminated structure as you have in humans and monkeys. The W cells innervate the C layers of the lower C layers, and the Y cells only innervate one of these C layers called C magno. So with large cells, these are the Y cells, these are W cells. This is a mixture of X and Y. If we look at the monkey, we see a similar lamination pattern with some details that are different. The parvocellular cells innervate the dorsal layers 3 through 6, and the magnocellular cells innervate the ventral layers 1 and 2. And the conio cells, the equivalent of the Y cells, terminate at the bottom of each of these layers. And that's where the conio cells are found. I mention this just because parallel processing, again, is a feature of visual processing and thought to be a feature of other processing as well. And it's curious to me that this was a very hot topic through the 70s and 80s and early 90s, and people don't pay much attention to this anymore, and I'm not sure why, because if we want to understand how sensory processing works, we need to understand more about how each of these separate channels is handled, particularly at cortical levels, and we don't know much about that. But again, here's a typical X cell in the cat. This is data from the cat, and Y cell. So even at the LGN level, there's different morphology for each of these channels, the same for the W. But another interesting feature, if we look at the lamination in the cat, is that the X and Y cells that are found in the A layers and this layer here represent the core pathway from the LGN to cortex. It's the W cells that represent the matrix pathway. Remember, these are the inputs that innervate mostly middle layers of cortex. This is the input that innervates mostly layer 1. And we see the same thing in the monkey. The parvo and magnocellular layers provide the core input. The coniocellular layers provide the matrix input. So these matrix cells are found even in the LGN. They innervate upper layers, and we know so little about what they're doing in cortex. I might add, in the monkey, you hear about LGN innervating just area 17, or primary visual cortex. That's true of the core input, but these matrix cells also innervate many extrastriate visual areas, and the function of that is not at all understood. So how do these parallel pathways operate through cortex? Do these pathways stay separate and analyze each of these channels of information independently? We don't know. Again, because research on this hasn't been very active in the last 20 years or so. But the following generalizations seem to be useful to think about. The two magno and parvo pathways converge into V1 onto separate layers. So even though they're both found in V1, the magno and parvo pathways are kept separate in separate layers, and they're processed independently. And there's a dorsal stream of cortical areas that is thought to be mostly involved in motion and location, and a ventral stream that goes out in the temporal lobe of cortical areas. It's mostly involved in fine detail and shape discrimination. And there's a generalization that the dorsal stream is mostly part of the magna pathway and the ventral stream more of the parvo pathway, although there's evidence for mixture at all levels. So this is a gross generalization to simplify things. Conio, we know nothing about. So that's it for parallel processing, which is a feature of sensory processing in thalamus and cortex. Any questions about that before I jump on? I'm jumping around here because there's no real organization of this. I want to say something about synaptic numbers. I think this has real implications for connectomics and presents some real challenges there. Let's consider the glutamatergic inputs that innervate a geniculate relay cell. We think of circuits in the brain in the following way. They can be divided into glutamatergic inputs, which we think carry the main information, and modulatory inputs like cholinergic and so on that are modulating those circuits, and GABergic that provide inhibition. It can be thought of as a different kind of modulation. But here we have two glutamatergic inputs that, as we'll see, have very different functions. If we want to ask the question, which of these inputs is providing the main information to the relay cell to be relayed to cortex, we can get an answer to that by studying the receptive field properties of each of these units. Because how a cell responds to its visual stimuli tells you a lot about what kind of information it's able to pass on to its postsynaptic targets. Well, the receptive field of the retinal output we know is the classic center surround organization. And these receptive fields are monocular, meaning only the eye of origin of the retinal axon can drive them. Stimulating the other eye will have no effect on them. So that's the information coming in. What about the information that might be coming from layer 6 of cortex? These receptive fields are more complicated. They have binocular receptive fields, so stimulating either eye in the appropriate area can cause the cell to fire. They have specificity not seen here for orientation, for direction, and for many other features. These are much more complicated receptive fields than these. If we want to know which of these is the input that is being used to inform relay cells, we could ask what the receptive field of the relay cell is, and it's the same as the retinal input. This has been known for decades, and this is why we say that the LGN is relaying retinal information to cortex. That's pretty clear and unambiguous. For that reason, we're calling the retinal input the driver input, because that's the main information that is being used here, which raises the question, what's going on here? Why do we have this other glutamatergic input with this kind of information that is not clearly seen in what's being fed up to cortex? Well, for a lot of, I don't have time to go into this, but we're calling this a modulator because it behaves very much like classic modulators, cholinergic and so on, with one important distinction. The classic modulator systems are very diffusely organized and seem to be related to overall behavioral state. The glutamatergic modulators are the only ones that have a great deal of topography. And we need topographic modulation for such things as spatial attention, adaptation, and so on. And only the glutamatergic modulators seem organized in a fashion to do that. Anyway, we do have these differences. Now we're going to see how these inputs are organized onto the relay cell. So we have two examples here. Data from the cat, but this applies to the rodent as well. We don't have data from the monkey, but hopefully, because it applies to cats and rodents, it applies to monkeys. We collapse the dendritic tree into a single dendrite, and it shows the inputs and where they're located in roughly proportional numbers. So we see cortical input out here, input from TRN axons out here, and closer to the cell body we have our retinal input and interneuron inputs. And remember, this is the triad that we see in glomerulus, and it's interesting that these are only related to X cells and not to Y cells. So Y cells have a very simple way of getting retinal input across to the relay cell, whereas in X cells you have this complex triad that may play a role in contrast adaptation. But what about numbers? The retinal input, the one that we think is so important, provides only about 5% of the synapses on the dendritic tree. The cortical input provides almost an order of magnitude more. These are the numbers for other inputs. If you just wanted to think about glutamatergic inputs, thinking that glutamatergic inputs are the main information route, and think of the brain organized in some sort of democracy for dendrites, and use connectomic data that would give you these numbers, you would come to the conclusion that what's being relayed here is provided by the cortical input because it's so much more than the retinal input. And the retinal numbers are very low and probably not very significant. Well, it's because we have functional data that we know that's wrong. This is the important input. And we know from synaptic properties that you can't get just from anatomy, that these provide very powerful synapses and these provide very weak synapses, which is part of the answer to this. What I'm saying is, if you just rely on synaptic numbers or even axonal numbers in pathways, you can be very badly misled. And I submit that you would definitely be misled if you try to use that strategy for thalamus. And I believe the same holds for cortex. That the driving inputs in cortex that are equivalent of retinal inputs are very small in number compared to the modulatory glutamatergic inputs. But numbers in cortex are much harder to obtain, and we don't have as good data for that. Notice also the dendritic tree and where the inputs are coming in. Jeff made the point a couple days ago that there's surprising specificity in the way an individual axon will target some cells and ignore other dendrites from other cells, even though they're physically as close. There's specificity in cell-to-cell contacts, but the specificity goes even further than that, because there's a cortical zone that's innervated and a retinal zone that's innervated, and they don't overlap at all. And furthermore, for inhibitory inputs, the TRN inputs are distal, the interneuron inputs are proximal, and they don't overlap. This is saying that the specificity for these contacts apply not just cell to cell, but what area of dendrites that are innervated. And the significance of this isn't entirely clear, but it is a very dramatic observation of the way these circuits are organized. Any questions about that? Yeah.
Would you remind us, in terms of correspondence of X and Y to P and M in monkey, which one?
Yeah, so X is equivalent to parvocellular, Y to Magnus. And when I say equivalent, I mean homologous, and that has implications for evolution. And W and conio are homologous. Yeah.
Can I just get a quick, like, how many neurons are firing between retinal input to V4? Like, is it like 3 or is it 5?
Retinal, I'm not sure what your question means.
Like if you have a photon hit a photoreceptor, retinal neuron fires, like how many steps are we talking
How many synapses do you have to go across?
Yeah, how many synapses?
Okay, so the retinal input, well, you know the retinal, you go from photoreceptor to bipolar cell to ganglion cell to geniculate cell to V1, and you can go from V1 to V4. You can go from V1 to MT, you can go from V1 directly to lots of areas. Another possibility is you can go from retinal ganglion cell to the conio cells, or W cells, and get directly to V4 from the LGN, but we don't know what that pathway is doing.
Do you know what that corresponds to in terms of timing, like roughly?
What's interesting is that we think of, this is just one example of why that's so surprising and hard to understand. V2 in the monkey is higher order than V1. But if you look at latency to visual responses, most responses in V2 actually happen before V1. And one explanation for that is that the axons that are innervating that pathway into V2 are coming through the magno pathway, which has much faster conductive velocity, whereas the responses in V1 are dominated made more by the parvo pathway. Beyond that, I don't think we know a lot about latency. If we get, there's another point I may get to, I want to bring that up again, because I think latency is an important issue, but to me very, very confusing. I'm not sure what it means. In fact, I'll get to it here. So now I want to talk about transatlantic cortical processing. And this is a new circuit that we're very excited about that 10 years or so ago wasn't known much about, but now we and other labs are studying it, and it goes as follows. I'm going to compare two thalamic nuclei here, the LGN and the Pulvinar. Now the Pulvinar is the largest thalamic nucleus. It's roughly 30 to 50 times larger than the LGN by volume, and the size difference here is important. We think both are visual relays. We know the LGN is visual because it relays retinal information. The pulvinar we think is visual because, remember I said cortex has about 36 areas that are visual areas. Pulvinar innervates all of them. So we know what the pulvinar innervates, but the question is, what is driving the pulvinar? So the LGN and the pulvinar both have the same non-driving input from local GABAergic cells, input from the brainstem reticular formation, descending input that's feedback from layer 6, that's modulatory, we think. The driving input from LGN comes from retina. The mystery for a long time is what the driving input to pulvinar was. The driving input in retina often occupies glomeruli with large terminals or proximal dendrites, and those were defined and described in Pulvinar decades ago. But the mystery is what the source of these synapses that were seen to be driving synapses. And now we know from research in a number of labs that the origin of this input comes from layer 5 of cortex. So we have something very interesting here. All thalamic relays get a feedback from layer 6 of cortex that's modulatory. But the pulvinar, in addition to that cortical input, gets an input from layer 5 of cortex that's organized in a feed-forward manner, not a feedback manner. And this pathway, functionally and anatomically, looks very much like retinal input to LGN. So what we think is going on here is that the pulvinar serves to relay information from one cortical area to another. So we're calling the LGN an exemplar of a first-order relay because it relays information for the first time, visual information in this case, to cortex. We're calling pulvinar an exemplar of a higher-order relay because it's relaying information that's already in cortex, but from one cortical area to another. Another way of looking at it is the first-order relay get their information from subcortical sources, from retina, from the medial lemniscus if it's somatosensory, from inferior colliculus if it's auditory, whereas the higher order relay gets its driving input not from a subcortical source, but from layer 5 of cortex. And this higher order relay that can relay information from a primary area to a secondary area can keep going as much as you like. There's lots of bandwidth here because remember, the pulvinar, the LGN just does this once. The pulvinar can do this for 30-some areas, and the pulvinar is 30 to 50 times larger than the LGN. So the anatomy here matches, at least in a crude way, to enable it to do that. Now, this first and higher order division, where the higher order relays are actually relaying information between cortical areas, is seen not just in the visual system, but also the somatosensory system, VPM and L are the first order relays equivalent to the LGN, relaying lemniscal information to cortex. The posterior medial nucleus is the equivalent of pulvinar. The ventral part of the medial geniculate is relaying auditory information from the inferior colliculus. That's the first order auditory relay. The dorsal division of the medial geniculate is a higher order relay, and so on. And when we look at thalamus as a whole, it turns out, as this which would suggest, that most of thalamus, whose function was kind of mysterious because we didn't know what information was being relayed, most of thalamus turns out to be higher order. And so we can ascribe a fairly straightforward suggestion for what most of thalamus is doing, like the pulvinar. It's relaying information between cortical areas. Now, these pathways exist, but what's curious is that there are also direct connections between these areas. And it's a very curious arrangement that seems to generalize, although we don't know if this is completely true in all cases. When there's a direct connection between two cortical areas, there's a parallel one that involves thalamus. A very curious arrangement. But this is based on a relatively small number of examples in the mouse, a few in cat, and a few in monkey. We'd really like to get many more examples to see how general this is, and this raises a number of questions. Before I get to that, there's another curious anatomical point. These axons that provide the input to be relayed, both for first and higher order, are branching axons. And they all send a branch to other structures in the brainstem, not just the thalamus. And many of these structures are motor centers, like the superior colliculus or the rubrous red nucleus, structures that are involved in supraspinal control. And I probably won't have time to say much about this. I'll just throw out the anatomical fact that needs some explaining. So this raises a number of questions. How common is the parallel organization of direct and transatlantic pathways for cortical functioning? Or how often do you get two areas of cortex connected just directly or just through the thalamus? We don't know. But the examples we have suggest that this parallel organization is the least common. What is different about the information that's going direct as opposed to through thalamus? Presumably different kinds of information are being sent through each pathway. And what is the difference? And why is one processed through thalamus instead of going directly? What's special about that information route that it takes a thalamic route? And finally, what is the significance of these branching axons? These are the four questions that we in other labs are trying to get a handle on. And that's pretty much where I can leave it for now. Maybe some discussion later, but yeah.
Some neuroscientists suggest that Pulvinar has a slightly different role or additional role that, for example, if we kind of prepare for a visual task or so, that it activates a small subset of cortical areas and also orchestrates maybe a common rhythm or so in those.
So Pulvinar can, because Pulvinar innervates all the areas, groups of Pulvinar, and we don't know what the mapping is from Pulvinar to each of the cortical areas, which makes it very difficult to study. If you imagine there are 30-some odd different maps in Pulvinar, depending on which cortical areas are both targets and input areas. Now, by this view, these Pulvinar cells are just relaying information they're getting from layer 5 of cortex, just like geniculate cells are relaying retinal information. They're not doing much processing here. But the processing that's significant is taking place in cortex. And you can imagine anything you want, that different cortical areas through their layer 5 outputs, which have already processed this important information, through Pulvinar are organizing different cortical areas. Another way of looking at this is that imagine that a cortical area innervates 10 other cortical areas directly and through thalamus. And the reason you send things through thalamus is you can block it here. And what's going to be important for how cortical areas communicate is which areas are connected by both and which areas are connected only by the direct route. And this is where Pulvinar can play a big role, because whether Pulvinar's gate is open or closed can determine what combinations of cortical areas are in contact and cooperating. Lots of possibilities, lots of questions, but those are just speculations. Yeah, Mike.
But is it known how patterned the activity is from the VRF? So is it sensible, or do recordings suggest that you can get specific shutting down and turning on of those purple inputs coming in from the particular firm?
Yeah, we think this is pretty global, that the control of the gating that you're talking about, we think mostly comes from the layer 6 feedback. But again, that's speculation. This is fairly new, and trying to understand how it all works, you can imagine all sorts of hypotheses that need to be tested.
So maybe to add to that question list, two questions. Can you say something about the different latencies of these signals?
Yeah, I'm glad you asked, because if you go from V1 to V2, that's a pretty short pathway. And if you go from V1 to V2, near the mapping of the fovea, that pathway is very short latency. If you go from V1 to V2 and in areas that are mapping 15 degrees out, it's a much longer pathway. So the latencies, we think there would be very different. But if you go down to Pulvinar and back up again, you have the same route, same latency. And that's interesting. And it's also those are the fastest conducting axons in the brain are the ones going down to Pulvinar and back up. So we think these transatlantic pathways, whether they're going from V1 to V2 or V1 to MT, which is very far away, have a constant latency, but the direct routes are all over the place in latency. I don't know what that means, but yeah.
Can I try something out? So suppose you have area 1. So its job is to represent the information, but also it also has to ask questions like, where's the red on my retina? So that to ask the question, then I have to pull the thalamus. And so now we go to area 2. Now area 2 has a question, so it has two ways to do it. You could translate the question into something that area 1 understands. Or you could just replicate the entire circuitry that has its thalamus in there. And so now, but area 1, its representation is now input. So that gets the that's sent to the area 2 thalamus. And so area 2 can ask its questions directly in the same way that area 1 asked its questions. And so on up to the other areas.
I'm not sure I followed that, but
it's just, but you do have two very different things you do. One is you build these representations of simple cells, blah blah blah. But having those is, you're not done. You're just creating a data structure that you can use to do things like you have visual routines a la Rosama, where you're constantly polling the representation that you have to figure out things. And so one way of doing that is, one school of thought is that the way you do that is you actually ask the thalamus, because everything has all this fine detail stuff down there.
that depends on what kind of circuitry you think, what kind of processing is going on in the thalamus, which I will say is an open question. The very simple way we're looking at it, which may be too simple, is that what's going on here is the same as is going on here, meaning there's not much processing. You have mostly a gated relay. And that means that this information that's being passed along is basically a copy of what's being processed through the layer 5 outputs in all of these areas. What you want to do with that, I think again, you can speculate and come up with all sorts of interesting hypotheses. The challenge is to test them. And we have our own hypotheses. We have a different view of what this is all doing, but it's speculation that is no better than anybody else's. But that's what we're trying to figure out.
Okay.
Are there any loops in this? So you have that, you said it's all feed 4, but it could be that area n feeds back to area 1. Is that?
There probably are loops, but they haven't been identified. And there's an interesting paper that Francis Crick wrote in the late 90s called the non-strong loops hypothesis, that suggests that you shouldn't have strong driver loops going in these circuits. We haven't seen any. Negative data doesn't mean a lot at this point.
I have a question about this concept of relay cell, which you have using quite often. So if one has a connection where one synapse comes into the relay cell, and then the relay cell forms a monosynaptic connection to some other cell, then I view it as a relay. But I guess actually a relay cell gets input from many neurons, like 50, 100 neurons. So therefore it's maybe not just relaying information, but integrating information through computation.
Right. So let's take the retina, for example. It gets convergent input in the cat and monkey from very few retinal axons, 1 to 6. But it gets lots of input from more than 100 cortical axons. But we think those are modulatory, and they're not providing the information to be relayed, and that's based on receptive field properties. Whether these Pulvinar cells are getting similar to the LGN, getting input from just a few layer 5 cells with very similar properties as you get here, and the rest of these inputs are modulatory, which plays an important role, but a different kind of role. That's one possibility. The other is that these cells may be integrating input from layer 5 of multiple areas. That would be a different kind of role that we don't see for LGN. That's a possibility. That was one of the questions that was also being asked, whether that sort of integration occurs in structures like Pulvinar. I think it's still an open question.
Are there clear anatomical subdivisions in the Pulvinar?
Yeah, so here's a big problem. Imagine you record from a cell in Pulvinar, you stick your electrode in there, get a receptive field, and now someone in another lab wants to replicate it. If we're right that Pulvinar has 36 areas that haven't been identified or mapped out, I can't tell the next lab exactly what part of Pulvinar is recording from. Was I recording from a cell that got input from V1 or a cell that got input from MT? And that makes it very difficult because we don't know what the map is. And that's one problem with Pulvinar, that people have done recordings, but each lab has a completely different set of results. You can imagine these receptive fields would be fairly simple because they're getting their input from layer 5 cells of V1. But cells getting their input from some parietal area could have very complex receptive fields. The other problem is people have made lesions in Pulvinar. Pulvinar abuts LGN, so everybody makes their lesion away from the LGN. They want to make small lesions, and the same problem exists. The lesion in one lab probably doesn't affect the same circuits as the lesion in another lab. So you have results of Pulvinar lesions all over the map. Some lead to blindness, some lead to very little. And it's a real problem because we don't have a good map of Pulvinar and a good way of knowing when you're studying a part of Pulvinar which of the circuits of cortex you're affecting.
And how much certainty is there that a given Pulvinar neuron is only receiving input from one area of cortex as opposed to maybe getting multiple areas of cortex?
I think that was the issue that these circuits are important at the single cell level. Pulvinar gets inputs from all these cortical areas. But we want to know how single cells are, whether they're integrating input from multiple areas, and we don't know the answer to that. Okay, yeah.
As far as I understand, the thalamus exists in essentially all vertebrates, including primitive ones that don't really have much in the way of cerebral cortex.
Right. That's a great question from an evolutionary point of view. There's a lot of debate about whether the structures that occur that have been defined in fish, birds, and reptiles are homologous to the thalamus or analogous. And I think in the bird, the data is best in the bird. I think the data in the bird suggests that what is described as thalamus really is homologous to thalamus. The question is, where's cortex if that's the case? And there are structures in the telencephalon of the bird that for years were debated whether were they homologous to cortex or were they basal ganglia. And only recently, using molecular techniques in the last 5 years or so, there was a definitive answer to that there are cells, individual cells in the telencephalon of the bird that are homologous to neocortical cells. But they're not organized in layers, they're organized in nuclei. So there's a group of layer 5 cells here, another group of layer 4 cells here. Beyond that, we don't know much. So does seem to be a thalamus, there does seem to be cortical structures, and there are pathways between them.
So you can hypothesize that the function is in some sense the same.
Yes and no. The whole point of evolution, remember, is to take a homologous structure and provide different functions for it. So saying they're homologous does not imply necessarily they have the same function. I think that's an open question. Yeah.
So based on the picture there, it seems like
I don't hear you, I'm sorry.
Sorry, so based on the picture there, it seems like all the transatlantic sort of processing sort of before. Is there any evidence for feedback?
Most of this evidence for transatlantic pathways comes from the mouse. There's some scattered evidence in the monkey and cat, but not those kind of details. So we need many more examples, and particularly nice to have more from the monkey. From the monkey, we have some evidence that V1 to MT has both direct and transatlantic pathways, and some evidence to V1 to V2, but even the evidence that exists in the monkey isn't as good as you would want that you have in the mouse. I think the hypothesis is that this is a general feature of cortex that applies to primates, but it has to be tested. It hasn't really been resolved it hasn't been resolved well outside of the mouse right now.
So my question was about whether there is any feedback. Sorry, so my question was about whether there is any evidence for feedback through the thalamus.
Oh, right. So that's I have this as a feed forward. There's actually, and it's something our lab is working on, whether there are feedback pathways that do the same thing. We think there are, but we don't have evidence. We don't have good evidence for that yet. So it may go both directions. And it's interesting that the feed forward pathways are all driver information bearing, but the input from Pulvinar to V1 is all modulatory, just like the layer 6 input to LGN. So there may be feedback modulation going through thalamus. Okay. Let's see where I am now. Oh, I'm pretty much done. So let me just go through these conclusions, and there will be some extra time for questions if people still have them. First, I talked about core and matrix thalamocortical systems. I'm bringing it up again, even though I don't have much to tell you about it. Just keep in mind that the matrix pathways do exist, and they're mysterious. We don't know much about them. And from a research point of view, anybody who's interested in this is a really rich area to study. Are there other types of thalamocortical projections? So I lumped these as two types called core and matrix. But there is some anatomical evidence. A neuroanatomist named Francisco Clasca from Spain has provided evidence that there are thalamocortical projections that don't fit neatly into the core matrix pigeonholes, and that there may be other kinds of fundamentally different thalamocortical projections. We've all focused on the core pathways with some knowledge that matrix exists, but this may be too much of an oversimplification. Details of circuitry are important. And I try to make the point with the example of the layer 6 feedback to thalamus, that it's one thing to say that the pathway innervates relay cells and inhibitory inputs to the relay cells, and it looks like it's organized as a direct, from a pathway point of view, a direct excitatory and indirect inhibitory circuit. But that probably isn't how it's actually organized, and to get at that at the real organization requires understanding circuitry at the single axon level, which is almost never done. And most studies, experimental studies, that study the functional organization of pathways either activate or inactivate whole swaths of a source input. And I try to show by example in the layer 6 feedback how that can be very misleading. It's a problem. I don't know there's a good solution to it, but I think you need to be aware of the problem. The other detail that's important, and this is a concern for connectomics, is that numbers can be very misleading. Again, if we take the LGN, if you just use numbers of synapses in the circuits, you come to the wrong conclusion about what relay cells were relaying, what kind of information they were relaying. And we have good evidence that this is true throughout thalamus, that the information coming into thalamus to be relayed to cortex, whether it's from layer 5, whether it's from retina, whether it's from the medial lemniscus, represents 5% or less of the total number of synapses. And that the other glutamatergic inputs from cortex, from layer 6, have an order of magnitude more synapses. And the tendency, I think, still of people when they analyze circuits is to use this democracy point of view that the more synapses, the more axons, the more powerful, more important. Frankly, I think that's wrong, not just for thalamus, but also for cortex. You have to identify the functional properties of the inputs, which right now you can't do just from anatomy. Hopefully, in the not too distant future, we'll have anatomical tags for the different kinds of glutamatergic input, driver versus modulator, so that you can figure out and really follow information flow in circuits. Another point is metabotropic receptors. They're dominant. I mean, they're not dominant, it's the wrong word. They're found widely through thalamus and cortex. And in cortex, we've estimated, in the mouse at least, that most glutamatergic pathways within cortex activate metabotropic receptors. It's a subset that doesn't, that's a minority. But don't pay too much faith in that, because while that's true in thalamus and we have good numbers for it, we're just starting to accumulate these numbers in cortex. What I think I can say with some confidence is that glutamatergic pathways that activate metabotropic receptors in cortex are sufficiently common to warrant consideration when you're modeling cortex, and this provides some very different functions. One other thing I may point out, so there's a long literature for plasticity that says that things like LTP that we used to think was dependent just on NMDA receptors, in cortex there are many cortical LTP examples that are dependent on NMDA receptors, on metabotropic glutamate receptors, and not NMDA receptors. So they probably play an important role in cortical circuitry, not just in overall function, but in plasticity. And one last thing I'll point out. All cells that we know of, thalamus and cortex, have voltage-gated conductances that can be turned on or off, depending on membrane voltage, that play a very important role for how the cell responds. Cortical cells, for instance, in layer 5 can burst or not burst, depending on whether a particular calcium conductance is activated or not. But to change the activation level of all these conductances, the sodium channel in the action potential is an example of one that doesn't do this. But almost all other conductances you have to change the membrane potential by a little bit, 5 or 10 millivolts in either direction to affect this change, but that change has to be held for at least 100 milliseconds to switch the channel from activation to inactivation. And inotropic receptors are too fast to do this. It's the metabotropic receptors, when activated, that can change membrane potential for hundreds of milliseconds that we think are key to controlling which ion channels are going to be inactivated or activated in cortical and thalamic cells. Most thalamic synapses, and we think this is true of cortical synapses, as I said, are not information bearing. Again, this gets to the point of parsing circuits based on total numbers can be very misleading. Parallel processing exists throughout thalamus. How far you can carry this in cortex is still, I think, an interesting question, but frankly one that's not a very hard issue at the moment. The existence of transatlantic cortical pathways we think is an important new way of looking at cortical function. These pathways do exist, details of what they're doing, and why you have a transthalamic route as opposed to direct routes, or I think I raised those questions. So I'm going to leave you with try on a higher level. We'll bring some art into this. This is a painting, a very famous painting in the Sistine Chapel by Michelangelo. And the title was Giving Spark of Light to Adam or something like that. I can't remember the title. But apparently the title was not Michelangelo's. He never named this. And the idea that God is giving life to Adam is something that Michelangelo never suggested. Now, Michelangelo was an interesting man because he was very interested in anatomy. Anatomy was something important for him for painting and for sculptures, obviously. He was a grave robber. He took as many bodies as he could to study them and dissected them, and that led to a lot of his artwork. But he was particularly interested in the brain. And he was probably the world's foremost neuroanatomist of his day. When he could get a brain in good condition, he took it out of the skull, dissected it, and tried to identify structures. The reason all of that is interesting is that if you look at this picture from a slightly different point of view, you realize that this is the brain. There's a pineal gland, there's a spinal cord, this is cortex. So the idea, another interpretation is that God is not giving Adam life, but giving him intelligence. And the final point I'd note is that God, sitting here in the brain, is sitting on the thalamus. That's his throne. And so he recognized that it was very important. Okay, I'll leave you with that thought.
Now I have a question.
Don't ask me questions about God, please.
Where did you get the idea about the brain? Did you read this somewhere, or did you come up with this yourself?
The what?
The brain, the image of the brain,
From,
did you just recognize it, or did you?
oh, there are other, you mean from Michelangelo?
The outline.
Yeah. What's your question about the outline? Oh, this is something that's not my idea. This is, there's a literature, let me just, since you asked, I'll give you another example here. I'll do with it here. Okay, so here's another one of his paintings. It's also in the Sistine Chapel. And if you look at this region here, here's the brainstem. There's the thalamus. And if you look more closely, this is, these are the two eyes, the optic nerves coming together at the optic chiasm. He embedded parts of the brains in many of his paintings, and he did it in a very sneaky manner. He never said anything about this, so all of this idea that he was putting brains in his paintings is fairly recent. Not mine. Other people have written about this, but I think that's kind of interesting.
Yeah. He was anticipating us.
Sorry?
He was anticipating us that one day.
Yeah, right. He was, in many ways, a man ahead of his times.
And Leonardo da Vinci did the same thing.
Do you have a question?
Yeah, just a very overall a general question. So you've given a lot of directions or research questions for the neurophysiologists in this audience. What role could the computer scientists play in this? And I think you did a nice job of really giving the dichotomy between functional and anatomical work, and I think the computer sciences could sort of really play a role in the functional aspect. So I just want to know what your suggestions to us.
Yeah, I think it's a great question, and maybe the point of this whole semester is to try to figure these things out. I think there are a couple issues I would raise, but I don't have really good answers for how they would be incorporated. One is that when you're modeling circuits, say in cortex, my understanding of these models is that if you have synapses involved, you have a simple alpha function that would be basically modeling what ionotropic receptors are doing in these circuits. I would like to see metabotropic receptors brought in. Having said that, I would also acknowledge that we probably don't understand them well enough to figure out how modelers would make use of that. But I think that to the extent that we really want to understand cortical circuitry or any other circuitry, the metabotropic receptors have to be understood and incorporated. The other thing I would say is when I see models, the ones that I'm familiar with tend to treat circuits, again, as some sort of democracy, that strength is based on inputs, and I think that's wrong. But again, having said that, I don't have a good answer for how you deal with that because we don't know how to identify one versus the other. But maybe models can be made explicitly with these two kinds of inputs to come up with something more interesting. I guess those would be my two main points.
I also wondered about this question, and I want to propose a hypothesis that I think almost all models that I'm familiar with, at least when they come from a computer science background, they really focus on a single computation operation, a single task, like visual processing. And so therefore, these models may not need a thalamus from the functional perspective. As soon as they would model now a system that can solve many different tasks depending on the environmental demands, then you maybe need a control circuitry which switches on and off particular parts of this larger circuitry. And then perhaps the thalamus would come in as a source.
Yeah, I haven't even suggested that the thalamus is an important part of modeling cortex yet, because to me, this may be very naive. I think models of cortex can be very well executed with just an input that comes from thalamus that may be turned on or off. But the issues I'm trying to raise is what happens within circuitry when you do two things. You bring in a different kind of postsynaptic response, and you have two very different kinds of strength of excitatory inputs. And the evidence from the mouse is that if you plot synaptic strength by the size of the EPSPs or by the probability of release, which is another way to measure it, the glutamatergic inputs are bimodally distributed. There's a small peak that has very high probability of release, shows depression and very strong EPSPs, and a larger, a more spread out one that is the opposite. And I don't know, I'm not sure how useful that idea is, but I think eventually, to understand cortex, those ideas should be incorporated.
We reconvene in about 30 minutes.
Oh, wow.